Hello and welcome to our third in the series of telling our stories in conversations between academics from throughout the university. The first two of these have been remarkably insightful. They've given us a real flavour of the kinds of work that our academics are doing, but also the kinds of connections that might link people who seem to be from disparate parts of the university. And today is no exception. We have two fantastic contributors. We have the return of Professor Graeme Earl from Archaeology, and we have Roxana Carare, who's new to this series, from Medicine. And again, this series is about trying to bring people together who might be of like mind, but of different experience, and see what might be a way of bringing those experiences together and finding out what the commonalities are, what the connectivity might be, and learning a lot along the way. So I'll hand it over to Professor Earl and take it away.
Okay, thank you very much. Maybe I could start by sort of saying a bit about me and what I mentioned last time when I was talking to Cheryl Metcalf from Health Sciences. So I'm in the Archaeology department, but I'm very interested in the application of digital technologies to cultural heritage generally. And one of the areas that I'm interested in is how you can stimulate different kinds of senses as a way of giving an impression of interacting with some kind of old artefact. So whether that's giving a visual impression or an auditory impression or a haptic impression, the touch obviously. And so in course of that conversation, one of the things we talked about was how these kinds of technology can be used therapeutically. So by giving someone a digital sensation of touch, it might be helping them in some medical or other way. And one of the areas that we talked about was dementia, where museum handling collections, so real objects, have been used to I suppose, help the lives of people with dementia. And I guess that's where us meeting came from.
Common denominator word.
Yeah.
So I'm Roxana Carare, and I'm a clinical anatomist. So what that means is I have a medical background, I teach anatomy, and that informed my research. And what's that got to do with dementia? A lot, actually, because one of the key aspects in anatomy is lymphatic drainage. We know that in cancers, metastases cancer spreads through these lymphatic vessels. Now, the brain doesn't have lymphatic vessels, and therefore it has an inability of dealing with waste, because one of the key functions of lymphatic vessels is to eliminate waste. So for a very active organ, like the brain, over the course of a lifetime, to not have lymphatic vessels, there must be consequences. And while I was teaching anatomy and reinforcing the fact that the brain doesn't have lymphatic vessels, I asked myself, what are the consequences of not having lymphatic vessels? And the person that then became my mentor is a retired professor of neuropathology, Roy Weller, and he showed me the first cases that I'd seen of Alzheimer's disease, and he said, well, look at the deposition of all of this waste in the brain. Mm-hmm. Then we started asking ourselves the question, could it be because we don't have lymphatic vessels? And here we are nearly 20 years later. We established that the deposition of waste products in the brain is specifically in the walls of blood vessels, and those tiny channels where waste deposit in the walls become compromised with increasing age, with risk factors such as high cholesterol, high blood pressure in midlife, and that sparked a whole program of research on why do these channels become blocked? How can we prevent that happening? And of course, because the blood vessels are key not just for supply of blood to the brain, but also now we know draining waste and lymphatic drainage off the brain, we're looking at ways in which we can investigate these channels in the living people. Because it's no good, it's very valuable, of course, for research to look at these brains after people die. But what can we do to prevent that happening, and how can we diagnose that very early? The problem is that these channels are about a millionth of a millimetre.
Okay.
And they're therefore very difficult to see with the imaging methods that we have available. But perhaps one of the most interesting things is that there are groups of people around the world that develop dementia very, very early and develop the accumulation of these waste products in these channels very early, age 30, early 30s. They're demented by their early 40s.
Right.
And of course, the question is why and what are the patterns? And I guess this is where your expertise and your background will come in extremely useful, because we've learned, for example, that in the Icelandic form of familial dementia, where very young people develop dementia, that form of dementia started in a family that imported the potato in Iceland. And therefore the environment and the early environment to which we're exposed becomes very important. So what is it that's different between the evolution of people in one area of the world with, say, a lower incidence of dementia, or where familial cases of dementia don't exist, and areas of the world where it does exist?
Well, so the tiny things that you were saying are indicative of this, and are those only present in the brain when you're studying it, or is there anything else that indicates that?
So those channels are in the blood vessels, in the walls of the blood vessels of the brain. However, the blood vessels of the brain come from a mother blood vessel in the neck that of course also supplies blood vessels to the skull.
Okay.
And that's where, of course, the blood vessels that are lining the skull groove the skull. Now, I've only looked at skulls from people that have died in the last 20 years, but I'm assuming that these grooves can be detected in skulls that are very, very old, thousands of years. What is the how old does the skull, or can the skull be, for you to see patterns of imprints, grooves on?
So we're using the kind of imaging that's done here. It might be possible to study. I mean, I know that my colleague Sonia Zakchevsky works with the anatomy department and also with the computer tomography centre here, the MUVIS centre here, to do analyses of I think there's some cranial things, but also things on teeth and various. So presumably, we've got the methods that would allow us to record things of that tininess, that size. And what you'd want to do then is apply that across a large population through time. So I would have thought that's, if it's not already done, that's interesting. I guess another thing that occurred to me when you were talking about the potatoes is that archaeologically we can find out quite a lot about diet, either through analysis of the bones themselves, or through kind of correlating information, so paleobotanical evidence, so indicating the presence of particular cereals, or, you know, so I was thinking about, I know of in some environments in Oceania where you've got populations subsisting almost entirely on shellfish, and you've got other populations who subsist almost entirely on taro, so something, you know, very carbohydrate rich. So presumably if you match that together with the skull evidence, you might see if there was kind of a consistent correlation between diet, other environmental factors, and the indicators you've identified.
I think that would be very valuable, because we'd be looking into the past to establish some patterns by which diseases have developed, but actually we can use that information to prevent it from happening. In a parallel universe, today's world where I'm working in, and of course you are as well. In the University of Southampton, we have the Barker hypothesis. David Barker was instrumental in establishing that diseases in an adult body may originate in utero while that body was in the embryonic state and fetal state. And we have a whole program of research looking at how diabetes may develop in adults because of the mother's environment. So with that framework, or within that framework, we examined brains from pups whose mothers were fed a high-fat diet or not. And in the pups whose mothers were fed a high-fat diet during pregnancy and lactation, there are changes in the morphology of the blood vessels of the brain and in their ability of coping with waste products. And we were very fortunate that very recently we were awarded some funding from Rose Trees Trust to examine these pups for a longer period of time to see whether these changes are maintained, worsened, or do they disappear if the pups are put on a normal diet with time. Although those are mice. They're mice because it's not ethical to take a bunch of ladies and put them on a high-fat diet. But and also it would be much more, even if we had women that came forward with a certain lifestyle and diet and they agreed for us to study the babies, it would be a very long prospective study. So I guess what I'm trying to say is that even in experimental conditions using mice, there is quite firm evidence that the diet of the mother, so the environment, impacts upon the development of the brain, and even more specifically, the blood vessels of the brain, therefore increasing the risk of stroke or the various types of dementia, not even necessarily just Alzheimer's disease. So that, of course, highlights the benefit of looking back in generations, establishing patterns so that we can influence and prevent this horrible disease occurring in the different populations that maybe are at risk.
Well, I could envisage there being populations where, we have the burial remains of a group that seems to be quite enclosed. It didn't have lots of external contact. And I suppose if at the same time you could correlate that against some kind of, their evidence for their diet and other environmental factors, you could infer, the kind of the matrilineal relationships and use that perhaps to do some kind of study.
So do we know now what populations were more exposed to, I guess what would be called in the modern times unhealthy diets? I'm assuming that the diet was more homogeneous. No?
No, I so certainly, to hold up my usual hand, certainly not an expert in this, but my understanding would be that you have a considerable degree of heterogeneity in terms of diet in the past. Firstly, on environmental grounds, so on the basis of what is growing and what is available locally, but then also on our understanding of the networks connecting people together at different points in time. So in the case of where I work a lot in Italy, in outside Rome at Portus from 2,000 years ago, you would expect there to be a very heterogeneous population coming from across the empire, but possibly in terms of the foodstuffs that were available to that population, they may not have actually been very diverse for perhaps status reasons. There could have been loads of different foodstuffs coming through and passing through the port, but not all of them would necessarily have been available to the majority of people living there. So I think we could probably pick and choose data sets where we have a really good understanding of that connection with the outside world and an understanding of the environmental determinants on the paleobotany and hence the diet.
I think that's very interesting because even while I was listening to you now, I was thinking that there are some colleagues in the United States that are working on how the lack of vitamin B predisposes to Alzheimer's disease. And with the collaborations that we have in place, we're demonstrating that the lack of vitamin B early in life again leads to changes in the morphology of the blood vessels. Whether that is also associated with the failure of clearing waste or not remains to be seen. We're in the process of analysing those data. But I think that the environment and diet are very important players when we discuss the risk of getting dementia, of getting Alzheimer's disease. I think that's something that is that remains to be explored thoroughly. Possibly what people don't necessarily know, because it's not widely advertised, is that apart from increasing age, which is the strongest risk factor for Alzheimer's disease, the other risk factor is possession of a certain genotype, gene, that carries cholesterol around the body. And that must have already even an anecdotal, but there is a lot of scientific evidence between the ability to transport the cholesterol around the body, the consumption of cholesterol, the metabolic rate of eliminating cholesterol and using it for different processes.
Right. Well, I think there certainly is work on diet and on the production of specific foodstuffs, so I'm sure that could be linked through to it. I think when you were talking about the last thing that kind of occurred to me from an archaeological perspective is not so much the environment that creates dementia or Alzheimer's, but the environment within which people who are suffering from that or exist. And so it was just occurring to me, I wonder if there are any archaeological, like material culture indicators for the presence of people exhibiting dementia, or the ways in which those people were looked after. And some of that might be, I guess, to do with the longevity of those people, you know?
Absolutely.
If it was identified as some kind of early onset form. And other ones might be even as, as broad as sort of artistic responses to people with those kind of differences.
And whether it was recognised as dementia to start with, because obviously there are different cultural barriers and cultural practices that will influence the diagnosis of dementia in different parts of the world and at different times in history. But one thing that I was wondering whether it's possible or not, and that's to bring in, we've still got space on the sofa for a mathematician maybe, to look at whether it might be possible to correlate the symmetry and the branching patterns, that's as far as my mathematics will go, of the blood vessels in the brain with the symmetry and branching patterns of the blood vessels that groove the skull now and before, and see if there are any correlations with the regions where dementia is more or less prevalent. So,
there's not much. Obviously, you can understand how we can think about how that could be done in contemporary data sets. In the past, if you wanted to look at the brain material, that would be cases where there's soft tissue survival, which is obviously very, very rare. It's under certain conditions that you do have survival with the soft tissue. So you might be able to do some kind of analysis.
We could look at whether, even if it's just looking at the skull, we could look at how over generations the branching patterns and the symmetry of the vessels that groove the skull, just the skull vessels, how has that changed in the skulls that are apparently normal and have stayed the same? Because that will definitely have implications on the development of strokes and dementia. because my mathematician colleagues are telling me that if the vessels branch in a symmetrical manner, they are better at coping with waste drainage. Whether they're better suppliers of blood to the brain, I don't know, but they're coping with the drainage of waste better. So those of us with symmetrical blood vessels that drain the brain will be better equipped at fighting dementia than others.
Okay.
So.
All right. Well, I think our time's up. But thank you very much. It was cool. It was cool.
So you talk about stroke and dementia. So so does that mean that if you have dementia, you're more likely to have? How are they connected in terms of that?
There are connections from every point of view. So a stroke generally means that a blood vessel is either blocked and therefore cannot supply that area of brain, or it bleeds and it therefore cannot supply that area of brain. As a result, that area of brain tissue dies because it doesn't get its blood supply to function, and dementia occurs just because of the loss of brain cells and brain function. However, in Alzheimer's disease, where there is no evidence of stroke, there is deposition of these proteins in the walls of blood vessels. And sometimes that deposition is very, very aggressive, very extreme, such that the whole of the wall of the blood vessel, the whole thickness of the wall is gone. It's just replaced by amyloid sticky waste. And therefore that vessel is under great risk, huge risk of bleeding, creating a stroke. So in most cases, the pathologists that examine the brains from people that have been diagnosed with vascular dementia because they have had a definite stroke where they were paralyzed or there were problems with speech, etc., when they examine those brains, they see the stroke, but they also see the amyloid in the walls of the blood vessels. What came first is impossible to determine. So there's a very close relation between them.
So you said that about the experiment with the mice fed on high-fat and low-fat diets. What was the actual outcome of that? Because I don't think you said which diet led to what type of outcome.
So where the mothers were on a high-fat diet during pregnancy and lactation, but the pups were on a normal diet, except of course they were breastfed by the mothers that were on a high-fat diet, the brains were abnormal. Specifically, the blood vessels of the brain were abnormal. Now, because of the lack of funding at that time, we were only able to examine those mice up to 15 weeks of age. So that's about a young adult. Whereas now we have funding to extend on that, and it would be very exciting to age those pups, keep their diet normal, and just assess the effects of the maternal diet from when they were in utero. I guess this has major public health implications because it's not just about what we eat, it's about, and for our own bodies, it's about what we eat and how that is carried on, or the impact upon the developing fetus or, future generations. And nobody's even looked at grandparents and diet, or even paternal diet. Yep. So,
I just had a question about the heritage side, and Graeme, you said something about there might be evidence of cultural heritage in archaeology, so the context in which people lived, or maybe some evidence artistically or of someone writing something down. But, and I'm going to cheat a little bit because we did have a discussion earlier about the physical evidence that might remain yet. So we're talking mostly about remains that might be centuries old, but is there a possibility that there could be some physical evidence still in situ or in collections around the world that could be re-examined of people who might have lived a long time who had dementia.
Yeah, so you mean the skull? Obviously, you've got massive collections of skulls across the globe, which could be studied from this perspective. I guess I'd be interested to know to what extent that had already been done. I mean, you've got a lot of collections where medical imaging has been undertaken, particularly kind of like a medical computer tomography. The problem there, I would imagine, would be the resolution. And so the issue with, because we talked earlier about CT, medical CT, looking for these kind of indicators, and the problem is, because it's not very high powered, the resolution, the ability to resolve these indicators is quite low probability or impossible. Whereas the kinds of imaging that we do on people who happen to be dead and happen to have been dead for a long time, it doesn't matter how powerful it is, and it doesn't matter if you're scanning the skull for 48 hours, 2 weeks, you can resolve those micro details. So I guess that's why it might be interesting to compare those two data sets. And also the possibility of looking at those small number of case studies where you have surviving soft tissue. So whether through preservation, the bog bodies, or preservation due to them being frozen, or wind mummification or deliberate mummification. So I wonder whether some of the soft tissue studies might be appropriate. I mean, so it may be that it's done, but it's as a result of the conversation, it'd be interesting to see, to check. And we definitely need to talk to say my colleague Sonia Zachewski, who I mentioned, so she would be able to fill us in rapidly on this after our discussion.
The sofa's big enough.
Yeah, the sofa's certainly big enough. Yeah, there's room for Sonia and your mathematician.
Definitely, I think, because in a single discipline we can't answer these questions, where we can just make some observations, but we cannot design the studies that will go across generations. I'm lucky to go from mother of mouse to pup of mouse, not how many thousands of years in the past. And unless we do this, we can't project to the future.
And I guess, the sort of the usual other contribution that archaeology might bring is thinking about the different ways in which society can respond to these kinds of differences. And they might be evident in material culture. Or it might be that by bringing in, as I know you've thought of, ethnographic and anthropological data, we can think about different ways of treating the condition, but also different ways of forming a society within which these people are present and in which they're important.
Yeah, absolutely. We might discover big lymphatic vessels engraved or grooving the skulls, and why did they disappear later?
Or how were they supported for long periods of time in their lives, which would suggest that they were in a society that valued that person and was protecting them.
Absolutely.
Anyone else? Well, I think it's a thank you then. Could everybody applaud and thank you our guests?